Hi. So we have also finished major chunk of this syllabus, and I think we are just left with 1/3 part of this course. And today we will begin with this host pathogen interaction in real sense. So today we are going to discuss the interaction between viruses with host, and you can understand that when virus infect the individual, then the innate immunity plays a very important role. So in today's session we will discuss about the innate immunity during virus infection. Most of things you are aware, but all those points which you are aware I will quickly move through, so that you will have a better understanding that during virus infection what is happening, you can put it in correct perspective. Okay, so let's begin this session. So you know that there are variety of innate immune component, right? You probably remember the previous session when I have discussed innate immunity. Okay, and there are several component of innate immunity innate immunity. As I can tell you again, so first is the physical barrier. Okay, another is chemical barrier or biochemical barrier. There is a microbiological barrier. There are cellular component and humoral component. Okay, so here I will mainly focus on the cellular component, and I will focus on how the virus is recognized. So when we talk about the recognition of microbial pathogen, in that scenario we discuss about this pattern recognition receptor. And since this the biochemical barrier, which is consist of variety of uh, enzymes and all those things, this is mainly common for most of microbial infection. So I am not going to talk about that. Okay, and in humoral component there are you know that there are complements and there are some antimicrobial peptide. So complements do play an important role against virus infection. They basically the complement get activated against enveloped virus, and they can disrupt those viruses. Okay, so I am also not going to talk about the complement, and I will straight away move to the cellular component. Okay, even I will not discuss about the microbiological barriers. Okay, that is not so uh, unique in case of virus infection. However, there is a pattern recognition receptor, and the sensing of this virus in various compartment of the cell, that is a very important, and the kind of responses they are inducing during virus infection, that is also very important. So here we will see some of the viruses which is sensed by various PRR. So here we will try to understand what component of which virus is sensed by particular PRRs. Okay. So first I will take you in general innate immune response, and then we will, as we move on the pattern recognition receptor along with viruses, you will understand that okay, these viruses are sensed by these PRRs. Okay. So PRR senses the viral PAMs, and this viral PAMs are various. It could be a nucleic acid, it could be a protein of the virus. Okay, and this sensing and the detection of the virus is could be the direct sensing of the virus. Okay, and another way is that it could be sensed indirectly. Okay, so now let's first discuss about the direct sensing of viruses by pattern recognition receptor. So viral signatures are, you know that the viral nucleic acid is a key signature of viruses, okay, because virus replicate in the host cell, okay, and since it is replicating in the host cell, so these nucleic acids are present inside the cell. In general condition, in healthy living cell, these viral the nucleic acid is not present in the cytoplasm or some unique location, for example endosomes or endolysosomes, but during virus infection these are basically present in these positions, and these nucleic acid is quite different compared to the host nucleic acid that I have discussed earlier in previous session, okay. So the long double stranded RNA, RNA containing triphosphate and unmethylated CpG DNA from DNA viruses, okay, which is mainly present in the DNA virus, and the genome, in case of DNA viruses the genome is DNA, which is quite obvious, okay. In addition, so these are the signatures, long double stranded RNA, which is not usually present in the healthy living cells, okay. The triphosphate containing RNA, that is also not present. The hypomethylated DNA is also not present. So these are the unusual distinguishing feature, okay, and that's why we call it as a PAMPs, okay. Additional factors also helps in determining the nucleic acid is from viral origin. As I told you, viral but not host nucleic acid are normally found in endolysosomes, okay. So generally in endolysosome the viral origin nucleic acid are present. So this location is also is a trigger for the activation of antiviral responses after recognition of this nucleic acid in particular location. Here you can see that there is a endolysosome and there are some PRRs that will sense and then that will induce, okay. And viral but not host DNA is present in the cytoplasm. So this is also unusual, okay, location of nucleic acid, okay. There is a indirect detection of virus by pattern recognition receptor, and this indirect recognition of virus is basically you know that viruses hijack the host cellular machinery, okay, and when they hijack and when they make their own copies, then the cell are under stress, okay, and then there will be a stress responses, okay. So that what I want to highlight. So this is also sensed by the pattern recognition receptor, and when this is sensed, in general the cell trigger their own death, okay, that we call it as a programmed cell death, okay. So here again I would like to say that alteration in normal cellular processes, of course, because they hijack, viruses hijack the host cellular machinery, okay, such as acute decline in host protein synthesis. Suddenly when there is a virus infection, there will be a almost no synthesis of host protein, that is a stress, okay. Altered activity of ion channel and ER stress. So that will also result to the ER stress, because you know that protein is synthesized over ER, okay, endoplasmic reticulum. So there will be a massive synthesis of viral origin protein, and that also cause the ER stress, okay. Alteration in ion channel, you can understand. So basically the virus skews the cell machinery for its own benefit, and since it is taking too much benefit, so cell also senses those things, okay. So these are the indirect sensing of virus by PRRs. However, it is not very well characterized. Still, it's a very active research area. Okay, so people are trying to understand how this, what is the molecular mechanism of all this thing. Okay, although we know the phenotype and effect. Okay, when virus is infecting, there will be a cellular stress, and that stress cell undergo the cell death. Okay, so this is known, but the precise molecular mechanism is not very well understood. Okay. So responses to the virus infection I have discussed earlier, but I will quickly go through so that you will put it in this perspective. Okay, during virus infection this is happening. Okay. So there is a innate antiviral mechanism, and you know that there is a production of type 1 and type 3 interferons, okay, which is one of key thing. Another is production of pro-inflammatory cytokine, and in addition there is a one more innate immune response is there, that is cell death, apoptosis, pyroptosis. Okay. So these are the key innate immune response against virus infection: production of this type 1 and type 3 interferon, pro-inflammatory cytokine, and cell death. Okay, that is also protective response, I have explained you earlier. Of course, when the virus is not cleared by innate immunity, that will trigger the adaptive immunity. Okay, we have discussed earlier, and this is basically the generation of antigen specific long lasting antiviral immunity, which is mainly mediated by cytotoxic T cell, which is CD8 T cells. Here there is a involvement of natural killer cell, which we consider it as a innate immune cell. Okay, so natural killer cell and cytotoxic T cell, basically they kill the virally infected cells. Okay, so now you can understand the one of the outcome of protective response is production of this interferons and pro-inflammatory cytokine and the cell death, that is also protective response. Okay. There will be a generation of neutralizing antibody. A lot of antibody will be generated, which will basically mask the viruses. So antibody can coat these viral particle, and then this will be phagocytosed. This is a one way. They can coat their those molecules which is basically needed for attaching with the target cell. Okay, so antibody, if the antibody will be there on those molecule which is basically needed for the attachment to the target cell, that will, so once the antibody will bind over there, then they are not able to bind with the target cell. Okay, so this is the way by which neutralizing antibody works. So all these mechanism, basically all this state ensures the inhibition of viral replication, viral gene expression, and virion assembly. Okay, so these are basically checking points. Okay, so now I will quickly show about the innate immune response against viruses. Here, as I told you, there is a type 1 interferon, which is basically consists of interferon alpha. It has a 13 member, and there is a 1 interferon beta member. Okay, there is a type 1 interferon inducible gene. So when this type 1 interferon is produced in a reasonable amount, then that will trigger the interferon inducible responses. Okay, and these responses are basically triggered by interferon inducible genes. Okay, there are just few examples, but this is hundreds of genes. Okay, one is that IP10 and rentis. There are so many chemokines and all those things are there. There is a type 3 interferon. It is basically consists of interferon lambda, and there are 3 members. Okay, so they are also playing very important role in checking the viral replication. inflammatory cytokine. There are so many inflammatory cytokine like TNF, IL-6, IL-1 beta, so on and so. So, all those things basically you know that type 1 interferon is basically checking the viral replication, and inflammatory cytokines basically they are kind of alarm system in the body. Okay, they produce and then they trigger the alarm in order to recruit the more professional killer cells at the site of infection. Okay. So, this is a all this response, okay, and there is a trigger of cell death, okay, and cell death is triggered by various other PRRs. Mainly it is those PRRs are derived from NLR member, okay, where AIM2 is also coming in picture, okay. I will discuss in a subsequent slide. Here I will show you the how this type 1 interferon and inflammatory cytokine works. I will quickly show, which is very easy to understand, okay. So, basically type 1 and type 3 interferons induces apoptosis of virally infected cells, okay. That is one of key innate immune response against viruses, okay, making healthy cells resistant from the virus infection, okay. They depresses the cellular synthesis pathway, that when if you treat the cells with type 1 interferon, there will be a decline in metabolic pathways in the cell, okay. That is why they are also act as an anti-proliferative, okay, and it is used in treatment of some cancer, okay. They prepare and activate professional immune cells for development of virus specific immunity, okay. So, whatever common immune response, if it is not able to clear, then they will start generating specific immunity, which is mainly taken place by B cells and T cells, okay. Inflammatory cytokine is kind of alert system, and this when this inflammatory cytokine will be produced, then there will be a recruitment of immune cell at the site of infection, okay. It could be systemic, it could be localized, okay. So, here I am just depicting, probably you will like that. Here you can see there is a normal situation, and in normal situation there is a virus infection. So, this virus is basically dominating over the host, okay. But once there is a virus infection, this virus basically sensed by the our cells, and then there will be a production of type 1 interferon, okay. And when there is a sufficient amount of type 1 interferon, the situation will be absolutely upside down, okay. Here you can see that there is a heightened immunity after the production of type 1 interferon, and which can easily take which will easily take over the virus, okay, and the individual start becoming a healthy, okay. So, this is for quick understanding, and you will remember for your rest of your life, because this cartoon kind of presentation will be much more easily rememberable, okay. So, now I will take up what are the various specific senses for the viruses. Here you can see that there are RLR pathway, NLR pathway, TLR pathway, and there are some another, okay. So, here you can see that there are cell intrinsic innate immune sensors, and these are the basically RLR, NLR, and there are some another sensor which is consist of AIM2 and P202, okay. And there is some cell extrinsic innate immune sensors, basically it is consist of TLR, okay. Here you can see that TLR is playing a very important role in recognition of viral or virus or viral signatures over the cell membrane and endosomes, okay. So, I will take you to each one quickly. So, first is RIG-I and MDA5 which is playing a very important role in virus recognition. Okay. So, here you know this RLR pathway, I am just showing you again. So, RIG-I basically senses 5' triphosphate, you are very well aware. Here this is a new information, besides Sendai and VSV, they can sense the influenza virus, hepatitis C virus, okay, Japanese encephalitis virus, okay, and this can also sense the small RNA encoded by DNA viruses such as Epstein-Barr virus. So, Epstein-Barr virus is basically a DNA virus, okay, and during replication they make a small stretches of RNA, and this small stretches of RNA can be sensed by the RLR, means RIG-I, okay. So, you can understand that the RLR pathways are very important against not only for virus infection, RNA virus infection, but it plays a also important role in recognition of DNA viruses indirectly, okay. You know that MDA5 also senses the RNA molecule, basically they senses long stretches of double stranded RNA, and they can sense the dengue virus, okay. MDA5 can sense the dengue virus, West Nile virus, okay, and basically these two sensors basically RIG-I and MDA5 basically sense this these viruses, dengue as well as West Nile virus, okay. MDA5 basically senses the picorna virus and can activate cytosolic synthetic double stranded RNA. I have told you previously, if you introduce the synthetic poly IC inside the cell, then that will mimic like a virus infection, and it is mainly sensed by the MDA5, okay. MDA5 recognizes large double stranded RNA structure, which is basically lacking 2O methylation, okay. 2O methylation is needed, okay, generated in cytosol during the virus infection, okay. I have discussed about the LGP2 molecule. Basically, this LGP2 molecule is basically potentiate the RIG-I and MDA5 mediated signaling. Originally, it was discovered as a negative regulator, but the physiological studies reveal that this is basically potentiating the RIG-I and MDA5 mediated pathway, okay. So, sensing of viral infection, okay, we have discussed about the, now we will discuss about the NLR and others, that is M2 and P202. So, this is basically, you can see that there is a, in M2 there is a domain, okay, known as HIN200 domain, and there is a pyrin domain in case of M2, and in P202 there is only HIN domain. Basically, this domain is binding with the DNA molecule, okay, and upon binding this makes one multi-protein complex, which we call it as a inflammasome, which I will discuss in subsequent slide. So, here you can see that there are various kind of inflammasome. There is a NLRP3 inflammasome which can be activated by various PAMP, and there is a AM2 inflammasome, okay. Cellular stress such as disruption of membrane integrity and extracellular ATP release from a stress cell. So, you can understand that when the other cells are under stress, which is already infected, and when these cells is under stress, they will release the ATP, and this ATP can be sensed by this NLRP3 inflammasome, and then that will induce the production of IL-1 family cytokines, okay, which is consist of IL-1 beta, IL-18, IL-33. Okay. Sendai virus and influenza virus, they can also activate the NLRP3 inflammasome. Besides this, the DNA viruses can also activate the NLRP3 inflammasome. So, DNA viruses basically not only activate the NLRP3 inflammasome, but it also activates the AIM2 inflammasome, which is AIM2 can bind with the DNA molecule. Okay. And upon this activation, there will be a production of IL-1 family cytokine. Okay. You can activate this inflammasome by transfection of RNA poly-IC, which mimics like a virus infection. Okay. And there is a RNA from reovirus and VSV. Okay. So, these can activate these inflammasome. DNA sensor or AIM2 inflammasome can also activated by the vaccinia virus. Okay. Now, I will take you to the TLR, which we have discussed. In mice, there are these TLRs are present, TLR 1 to 7, 9, 11 to 13. Okay. So, only 11 TLRs are functional. In human, there is a TLR 1 to TLR 10, and only 10 functional TLRs are present in humans. Some of these TLR basically express on the cell surface, okay, like TLR 1, 2, 4, 5, 6, 11 and 12. Okay. In endosomes, there are 4 major TLRs which is present, that is TLR 3, 7, 8 and 9. Okay. So, endosomal TLR trafficking and processing. So, there is a one very important molecule which I have not discussed during TLR in previous session, but there is a one very important molecule is there which we call it as a UNCB93B. Okay. So, here there is a UNC93B. So, this molecule is basically expressed over the endoplasmic reticulum. Okay. And this is very much needed in order to take this newly synthesized TLR 3, 7, 9 to the endosomes. If you delete this gene, the TLR 3, 7, 9 will be not translocated into the endosome or endolysosome. Okay. And I think I have explained you these TLR do not sense the PAMs directly. Before sensing, they undergo proteolytic cleavage by family of proteases, which we call it as a cathepsins. Okay. So, if there is a mutation in this molecule, that is UNC93B, it is 93B1, then there will be a severe HSV encephalitis will happen. Okay. And this severe infection is quite complicated to control. Okay. So, you can understand there is only 2 patient so far we know, maybe more will be there, and if there is a mutation in this gene, then this will hamper the all TLRs which is translocated into the endolysosome. Okay. Endosomal TLR in virus recognition and signaling, I will talk now this. So, there is a TLR 3, 7, 8 in case of human and TLR 9. Okay. So, TLR 3 basically plays a very important role in sensing DNA viruses also, and these DNA viruses basically produce double stranded RNA by convergent transcription of their genome. Okay. What do you mean? So, there will be a promoter, 2 promoters will be there, and this promoter can, since it is present on 2 different strands, so this can make a RNA molecule, and that RNA molecule eventually become a double stranded RNA molecule, and this double stranded RNA molecule will sensed by the TLR3. Okay. So, there is a study that dominant negative mutation, so not functional, you can understand dominant negative that the TLR3 will be not functional for time being, okay, suffer from neonatal herpes infection, okay. Herpes is a DNA virus, but if there is a mutation in this TLR3, then that will result to this neonatal herpes infection. TLR7, okay, so human PDC and DC basically express this TLR7 and B cells also, and it is sensed by the influenza. Basically TLR sense the influenza, Sendai virus and VSV, okay. Mouse TLR8 is expressed by myeloid DC and monocytes, okay. TLR7 and 8 recognizes single stranded RNA, and the recognition component is uridine and ribose, okay. In single stranded RNA, this uridine and ribose is sensed, okay. Okay, so both this uridine and ribose is needed for sufficient stimulation of TLR7, okay. Viral fusion and or uncoating and endosomal acidification is required for TLR7 dependent recognition, okay. TLR8 deficient mice develop autoimmunity due to hyperstimulation of TLR7, okay. These are some additional point about these TLRs, okay, which is more specific to the viruses, okay. There is a TLR9. So, TLR9 is basically expressed in PDCs and B cells in human, and mice expresses TLR9 in PDCs, B cells, macrophages and conventional DCs, okay, and senses the viral DNA and bacterial DNA. TLR9, if you remember, TLR9 senses CpG region in DNA which is hypomethylated, okay. So, senses viral DNA and bacterial DNA sequence containing hypomethylated CpG motif. It is derived from, there is example, it is derived from HSV1 and HSV2. And recent study showed that TLR9 recognize the sugar base backbone, okay, 2-deoxyribose of phosphodiester DNA irrespective of CpG content. So, this is a more deeper insight for recognition of the DNA in case of TLR9, okay. So, we were thinking that okay, CpG motif is sensed, but this may be also true that this sugar base backbone and 2-deoxyribose of phosphodiester DNA is the key component which is recognized by the TLR9, okay. So, with this I am stopping here, and in next session I will talk about the some interferon independent mechanism, and in that session I will discuss about some of my work also, and I hope you will like that how this interferon independent processes are also playing very important role in controlling the virus infection. Thank you.